Welcome to MOOC NPTEL course on bioengineering, an interface with biology and medicine. Last week we started discussing about why biology is so important for engineering discipline. I try to give you various examples in which way we can see that the bioengineering has started making a huge impact in many applications. Then we kind of talked about basics, especially the life properties, the cell, different cell organelles and their function. I also tried to provide you a clinician's perspective by interaction with Dr. Ali Asghar Moyadi to give that in which way clinicians are also looking at various engineering solutions, the technological solutions for various medical problems. So, this week we are going to talk mainly about the DNA and DNA tools and how biotechnology has started making impact by knowing the DNA technologies. So, we are going to talk about the various technologies involved in doing the molecular biology research, especially polymerase chain reaction, different gene cloning processes. Those things we are going to cover in this week, but I thought it will be important before we start going into detail of those technologies to first introduce you again with the nucleic acids and central dogma. So, that is the theme for today's lecture. Although I realize that you might have already studied this in your earlier courses in other classes, but just kind of refreshing you about some of the concepts for the nucleic acids, gene and operon, and then I will talk to you about central dogma and in which way it is important in the omics era. And then these concepts, how we can try to utilize these understanding. Then in the next set of lectures in this whole week is going to utilize the DNA technologies and going to illustrate you in which way we can do research in these areas. So, let us first start with the basic concepts of nucleic acid, which are the basic components of DNA. So, the main function of the nucleic acid is to store and transmit the entire genetic information, and there are two specific classes of sugars based on which the entire classification happens for nucleic acid, which is DNA or RNA, right, deoxyribonucleic acid, DNA, and ribonucleic acid or RNA. So, the major constituents of the chromosomes which are located in the nucleus of a cell is the DNA, which constitutes the genetic material, whereas the ribonucleic acid or RNA is the functional molecule or the working copy of DNA, which then participate in the process of protein synthesis. So, from DNA to RNA the process of transcription happens, and then from RNA to protein process of translation happens. Let me now show you again and more so refresh you about the DNA structure and the component, the nut bolts involved in making DNA structure. So, for example, there are 4 bases, the monomers which constitutes DNA, and these are the structures shown on the screen, which is cytosine, thymine, adenine and guanine, ATGC. Now, cytosine and thymine, these are 6 membered ring with 2 nitrogen, which is having the pyrimidine ring structure, or the adenine and guanine, they share the structure with the purine ring, and which is pyrimidine plus addition of an imidazole ring. Now, uracil is unique because that is found in replacing thymine in case of RNA. So, you can look at the structure of uracil here. Now, let us talk about briefly the sugars. There are 2 sugars involved, one is the ribose sugar and another is the deoxyribose sugar. Both of them have the pentose sugar backbone, and now on the second carbon you can see there is a hydroxyl OH group there in the ribose sugar, whereas in the deoxyribose it is hydrogen. So, that is the difference between the ribose and deoxyribose. And then third important component is the phosphate chain, which is joined with the on the C5 carbon with the hydroxyl of the sugar, whether in case of ribose or deoxyribose. And then because of this phosphate group, these nucleotides, they are negatively charged, and that property is heavily used in the DNA electrophoresis. So, now let us kind of again look into these individual components. We talked about the bases, we talked about the sugar, and we talked about the phosphate, right. These 3 components together form the nucleic acids, and let us look at some terminology. For example, nucleoside, when you are combining a base and the sugar form, that together gives rise to nucleoside. or when we add a base, sugar and phosphate chain, that is nucleotide. And then when you are combining many nucleotides which are actually joined with the phosphodiester bonds, that is known as nucleic acids. So, a nucleotide is a subunit of nucleic acid which consists of the nitrogen containing bases, which is having the 5 carbon sugar and the phosphate group. So, again the structure is shown you here, which is for a nucleotide. So, now in this way, now you can easily decode the entire DNA structure, which is straightforward. Now we have good understanding that in which way nitrogenous bases, the sugar phosphate backbone is constituting the DNA structure, but of course it was not known earlier, and the scientists Watson and Crick, they get the credit for deducing the structure of the DNA and their how these are, the two strands are arranged in a helical form, how this is intertwined, and the sugar phosphate backbone lies on the outside, whereas the bases are inside. And then the base pairs are specifically forming the bonds, which is hydrogen bonds between A and T and G and C bases, right. So, there was always that a quest to elucidate the structure of DNA, and many scientists started working in that area and try to find out in which way the DNA structure is made. So, the Linus Pauling, he made a hypothesis that there are 3 chains which are twisted around each other and they form some sort of rope like strands, and that could be how the DNA structure is made. So, then Wilkinson and Franklin, these scientists, they provided X-ray crystallography data, and then they found that nucleotides are 3.4 angstrom apart in the chain, and the structure repeats are found at the 34 angstrom interval. So, that gave much more clarity for the the structure of DNA. And then a scientist Chargaff, he provided the some basic rule that the A components are going to be equal to the T components, so percentage of A equals to percentage of T and percentage of G equals to percentage of C base pairs. So, this summarizes the kind of what we have discussed, the structure of DNA, in which way adenine and thymine they form the 2 hydrogen bonds, and guanine, cytosine, they form 3 hydrogen bonds, and the structure of DNA decoded by scientists Watson and Crick, for which they were awarded Nobel Prize in 1962. Now, then how DNA makes its multiple copies. So, the DNA duplication or replication of DNA, that is another interesting concept. So, the DNA double helix is actually starts unwinding at the replication fork. So, now 2 single strands are produced from this double helix DNA, which serves as templates for polymerization of free nucleotides. Now, DNA polymerase it starts polymerizing these nucleotides by addition of some new nucleotides to the 3' end of the DNA chain, and now from the same DNA now the 2 copies of DNA is made, and as you can see that from the template S strand, now we have new strand being synthesized, and now we got 2 DNA molecules as a process of replication. So, there are different theories which are involved in the DNA replication. I will talk to you about the DNA replication and some of the classical experiments done in some other context later on, but just for the timing I thought to just give you the feel of that how DNA copies are being made, and why it is so important for us to understand DNA structure, because the DNA is so much fundamental to our life, to our hereditary information, that how DNA structure is found and how it is compatible with all any possible sequence of bases. That is very important, I think, for us to appreciate and understand that the sequences of bases along any DNA strand, they act as a very efficient means to store the genetic information. So, knowing the DNA structure becomes very crucial, and this DNA sequence actually ultimately determines the sequence of the ribonucleic acid, and eventually the proteins are formed from that. So, these sequences of bases along with one strand, they are completely determines the sequence of other strand, and then they are going to dictate the RNA formation and the protein formation. So, after reviewing some basic concepts of DNA and its DNA structure, let us now think about a question about what is a gene, right. The concept of gene has actually evolved through the history of genetics, starting from the scientists like Mendel, who was thinking about, these are some hereditary factors which are, he did not know about gene, that these informations are passing from one to next generation. Probably there are some factors which are involved which are having these information stored. And then Morgan kind of provided some further experimental evidences that these hereditary units are actually located on the chromosome. So, many scientists have contributed in the journey, and now finally we know that what we consider as gene is actually a discrete unit of inheritance, or you can also define that as a region of a specific nucleotide sequences in a chromosome, or you can say it is a DNA sequence that codes for a specific polypeptide chain. So, if you just want to get a broad overview of gene, I think we can summarize it is a region of DNA that can be expressed to produce a final functional product. It can be a polypeptide or it can be an RNA molecule. So, this is how you can think about gene, one of the discrete unit of inheritance which is providing this kind of functional information. Now, organization of a typical eukaryotic gene is really complex and having many processes which are involving to shuffle from the DNA to make the RNA, but kind of this slide try to illustrate you that we have various exon regions and we have various introns. Now, in the process of alternative splicing, in which way now these introns are removed and the coded form, the exons, are coming together to give rise to the functional RNA molecule. So, there are multiple control elements which are actually associated with the eukaryotic genes, and these are the segments of non-coding DNA which help to regulate the transcription by binding to the certain proteins. The concept of lac operon becomes very crucial. In this model you can see that in the if you have allolactose, which is an isomer of lactose sugar that de-represses the operon by inactivating the repressor, and in this manner this enzymes for the lactose could be utilized and then it can be further induced. So, I am going to show you this in one of the animation and to explain you in much more detail.
In prokaryotes, transcription by RNA polymerase can take place with the help of an activator protein. However, in the presence of a repressor molecule, the binding site for RNA polymerase is inaccessible, due to which transcription does not occur. In the ground state, the repressor does not remain bound, because of which the gene is turned on. The lac operon consists of a group of genes that are responsible for transport and metabolism of lactose sugar in certain bacteria like E. coli. This operon is under negative regulation by the LAC-I repressor protein. In absence of the inducer, the tetrameric repressor binds to the operator region, thereby preventing transcription by RNA polymerase. In presence of the inducer binds to the repressor protein, which then prevents it from binding to the operator and therefore allows gene expression. An inducible system is off in its ground state and must be turned on by an effector molecule, which is known as the inducer. In positive regulation mechanism, however, the inducer binds to the inactive activator to produce the active activator molecule, which in turn facilitates binding of RNA polymerase to the promoter to turn on expression. In the negative regulation mechanism, the inducer binds to repressor and prevents it from binding to the operator region. This allows RNA polymerase to proceed with transcription by binding to the promoter. The ground state in case of repressible system is on. It has to be turned off by an effector molecule, which is known as a co-repressor. In positive regulation, the co-repressor binds to the activator molecule and prevents its binding to the promoter region, thereby turning off gene expression. In case of negative regulation mechanism, the co-repressor binds to the inactive repressor molecule and activates it, thereby preventing gene expression. The lac operon consists of a group of genes that are responsible for transport and metabolism of lactose sugar in certain bacteria like E. coli. This operon is under negative regulation by the LAC-I repressor protein. In absence of the inducer, the tetrameric repressor binds to the operator region, thereby preventing transcription by RNA polymerase. In presence of the inducer binds to the repressor protein, which then prevents it from binding to the operator and therefore allows gene expression. Lac operon also undergoes positive regulation by means of the cyclic AMP cap system. Glucose is a preferred energy source for bacteria, and if both glucose and lactose are present, beta-galactosidase enzyme, which metabolizes lactose, is not synthesized. High glucose levels prevent synthesis of cyclic AMP, which is essential for binding to the catabolite activator protein. This protein facilitates transcription of the lac operon. When glucose levels are low, cyclic AMP is produced, which binds to the scap, which in turn binds to a distal part of the promoter region and facilitates transcription.
But this kind of slide illustrates you the broad model of lac operon and in which way it regulates the synthesis of inducible enzymes. Okay, so now let's move on to thinking about from the cell where we can find the DNA, right. So let's say human body is made of billions and trillions of cells, we have discussed in the last class as well, and then each of those cells is having these the nucleus which contains the genetic material. Now each cell contains 2 copies of these chromosomes, and now these chromosomes, if you expand further, you can see the long DNA molecules, even the genes and the functional region of DNA we can see over there. And then now you can see the tiny picture of how these DNA molecules, if you think about the cells, each cell having the nucleus, having the chromosomes, having the gene and their the DNA part. So this is a really tiny bit of the molecule present in the cell, but that is so crucial, which dictates all the hereditary information. And its kind of packaging in this cell becomes very crucial as well. And again, to refresh you from the previous lectures in the cell context, if you think how the nucleic acid contents are so tightly packed inside the nucleus in a small area with very intricate binding with histone proteins. So these histone proteins and these DNA molecules, they form these nucleosomes, and these nucleosomes together are packed to form the chromatins. So this is how these particular packaging happens inside the nucleus. And let's thinking about eukaryotic genomes, how they are organized into the chromosomes. I think the knowing about the histone proteins becomes very crucial. So these histone proteins, as I mentioned, H2A, H2B, H3 and H4, they are positively charged proteins which could interact with the DNA molecule which is negatively charged, and that actually helps to compact the DNA, and these nucleosomes could be seen like the beads on a string. So this is how you can think about from DNA to chromosomes. A chromosome consists of a DNA molecule which is packed together with the proteins, and now these chromosomes could be seen which are having these bead kind of structures, right. Alright, so human having 23 pair of chromosomes, 22 autosomes plus 1 pair of sex chromosomes, X or Y chromosomes, and a process known as karyotyping, where you want to look at the pattern of each of the chromosomes, tells us about the is the pattern of these chromosomes are normal, or is there any abnormality can be seen. And for many disorders, like especially chromosomal aberrations can be found, and that actually helps us to deduce if there is some sort of syndrome is present, like Down syndrome, or some sort of the issues with any other chromosomes abnormalities are there. And even for sex determination, people look at the X and Y chromosomes and their patterns. So, this image just shows the color painting of these various chromosomes, but ideally it shows us the organization for the whole genome. Alright, so there have been many discoveries which have contributed immensely to the field of overall DNA related discoveries, which have contributed entirely from genetics to genomics areas. I am going to show you a couple of milestones discoveries, although we are going to talk in much more detail about many of these fundamentals and their applications in subsequent lectures, but just to kind of refresh you and bring you to the scale, starting from Gregor Mendel, the father of genetics, 1822 to 1884, a lot of elegant experiments being done on the pea plant, which gives us the basic idea for how various laws of hereditary are governed. And then with the ideas for discrete factors which Mendel mentioned, the genes are actually going to transmit characteristics from one generation to the other generation. Over the period, then we had the discoveries by Watson and Crick, which illustrated the structure of DNA, and but the initial part from 1865 from Mendel is gets credit, and Mendel is known as the father of genetics for his contribution. Then as you go on the time scale, Sturvant he made the first linear map of the genes in 1913, and then came the Watson and Crick contribution for double helical structure of DNA in 1953. Then scientists Nierenberg, Khurana and Hawley, they first time mentioned the genetic code in 1966. Scientists Cohen and Boyer, they developed recombinant DNA technology in 1972. And then Sanger, Maxson and Wilbert, they developed DNA sequencing methods in 1977. Then 1983, the first human disease gene was mapped with the DNA markers, especially in the disease of Huntington disease was shown. And one of the milestone technologies, polymerase chain reaction, was invented in 1985. And then Human Genome Organization started an ambitious project of knowing about all the human genes in 1988. And then while those things were happening, we started knowing more about the cell, about cloning, about development process and reprogramming, and that eventually culminated into the cloning of an animal, Dolly, by Ian Wilmot in 1997. And that is another one of the scientific fiction and kind of story in which one could produce a cell or organism with the same nuclear genome as another cell or organism, and Dr. Ian Wilmot of Roslin Institute, they cloned this sheep Dolly, which was a major accomplishment that time. Then human genome sequence projects were getting completed in the years 2001, 2003, and then first draft of the human genome map was presented in cover page of Nature and Science. And those projects actually help us to really try to get a bigger picture of what is happening inside the entire human genome, what are all genes present there, and it was first most ambitious project to really understand beyond moving on to the single gene and looking at just the characteristics governed from each gene, that what is happening in the entire genome and all the gene, how they are governing the function. So that kind of was a big accomplishment, not only to understand the gene, but also in the scientific community, in which way we are able to now work for understanding all the molecules of life, for example, omics molecules. So this brings to the second part, which is thinking about central dogma. So now we have studied about DNA, which is the genetic blueprint, and just imagine that in a cell, now you want to first of all you want to know that where the DNA is, and that DNA is going to make the molecular photocopy, which is RNA, and that is like the functional molecule has to be initiated, and from those RNA molecules the proteins has to be made. So now just use the same analogy for, let us say, making a building. So, let us say we are in Mumbai, in Powai area in IIT Bombay, and we want to make a building, a campus here. So, from the map now you know that where is the DNA, the genetic blueprint, and in that area then some contractors will come and then they will try to make a map that where the building has to be made. And then the proteins or the building material will come, which is going to be like the mortars and bricks, which is going to create that building, which is like the engines of biology. So, this is something which helps you to provide a good analogy in which way DNA to RNA to proteins, everything is crucial, but in which way the proteins are much more direct information they are providing, much more functional molecules, while the genetic blueprint comes from the DNA. Now, let us kind of briefly refresh the classes of RNAs. We have the messenger RNAs, we have transfer RNAs and ribosomal RNAs. Messenger RNAs, they provide a template for protein synthesis for or the translation process to happen, but they are very less abundant, only 5% population is there for the mRNA, where the transfer RNAs, they are the carriers of amino acids and activated form to ribosomes. Then the ribosomal RNAs, they are the major component of ribosomes. They provide catalytic and structural roles, and they are actually the most abundant among the RNA population. So, RNAs, they are involved in the protein synthesis process. Again, this is an interesting but the complex subject which needs much more full lecture. But just to brief you, the mRNA provides the long sequence of nucleotides which serves as a template for the protein synthesis to happen. Now, the tRNAs, they are involved in the protein synthesis, but that binds an amino acid at one end and the base pairs with an mRNA codon on the other hand, and then that serves as an adapter that translates mRNA code into a sequence of amino acids. Then rRNA, it forms the central components of ribosomes. It also plays catalytic and structural roles in the protein synthesis process. So, transcription and translation, again there is a lot of fundamentals involved in understanding these processes. This slide just kind of gives you illustration that in which way from DNA the RNAs are being formed in the process of transcription, and then from there in which way various amino acids are formed and the translation process happens to generate the proteins. So, this is we will talk as we go along, but I just want to convey you that there is orderly and unidirectional flow of information happens in the cell as a part of central dogma, and that actually that information is in the base sequence of the DNA, which flows from DNA to RNA to the protein. And this is what we say the central dogma, which involves two important steps of transcription and translation. And of course, you are also aware that there could be reverse transcription as well, where the information could also flow from RNA to DNA, and that is also very crucial for many biological phenomena to happen. An important thing why from the same gene we still see different type of RNA forms and multiple protein forms becomes very crucial to understand, and that is actually being dictated by two important phenomena. One is alternative splicing. So, in alternative splicing, that is a process in which the exons or the coding sequence of pre-mRNA, they are produced by the transcription of a gene, and then they are combined in different ways during RNA splicing. So, what happens then, the resulting mature mRNA, it gives rise to different protein products as a part of translation, and then they are the isoforms of one another. So, now you had a single gene, but actually that can give rise to multiple RNA forms and different protein products. So, as you can see in the picture from the one of the pre-mRNA, we have mature mRNA A and mature mRNA B, they are being formed, and they give rise to the red color protein, protein A, or the green color protein, protein B. Then after the proteins are being synthesized, then further modification may happen at the protein level, and that is known as the post translational modifications. So, many proteins undergo post translation modification at some of their amino acid residues, and some molecules could be added, for example, sugar moieties as a part of glycosylation, or phosphate moieties as a part of the phosphorylation, or it can be hydroxylation, methylation, alkylation, acetylation, and many kind of modifications may happen, which makes the protein very different functionally. And that is where studying the RNA molecules or studying protein molecules provide much more functional information, because many of these modifications are actually quite relevant thinking about the actual physiological questions, right. So, this information what I just conveyed you looking at a central dogma, all the genes, then the transcripts and the proteins, now scientists are trying to study in much more totality. For example, can we study all the genes of a given organism, of a given system? Let us say for human we have now idea for the entire genome, and that we say the human genome project or human genome sequences are available now. And similarly, do we have idea for the entire human transcriptome or human proteome, and that can help us to really understand the system much better as compared to thinking about just one or two protein at a time. So, then the omics understanding, this whole field is known as omics field, which aims to look at all the molecules present in a given system, and then this information could be very valuable and useful for the patient's treatment and patient therapy. Think about the personalized medicine, which is an area upcoming right now, where intention is to look at these biomolecules from a given individual and use that entire information for their treatment. So, whether you think about having the diagnostic test, or think about the integrating these personalized interventions, or optimizing them over the period for the treatment modalities, all of them requires good understanding of these basic biomolecules, and if possible the technologies to understand them at the omics level. So, in summary, today we just talked about the basics of nucleic acid, especially the DNA and RNA. We also tried to cover briefly about histones and in which way DNA are being packed. Then I try to convey you that in which way central dogma flows the information from DNA to RNA in the process of transcription, and then protein in the form of translation, and then in which way the new emerging field of omics technology is able to is actually aiming to understand the complex signaling pathways which could be involved in the biological system, and that may lead us to do better cure at the personalized medicine level. So, these are all some of the basics fundamental. Of course, to know more in detail, you have to either take some more advanced classes, or you have to take more specialized biological classes, but our intention was to give you some overview to refresh you about these biomolecules. And now we are set to talk to you about various advances which are happening in the DNA tools and biotechnology area, and that will be the main focus for the next of the lectures for this week. Thank you very much.